All right, so now we're going to get finish up section 5.1 in chapter 5. So we just went through and talked about dominance and the three major forms of dominance, so complete, incomplete, and codominance. So just thinking back to the principles that we learned about in chapter 3, dominance is really going to be determined by the way alleles interact with each other. Okay, so and that interaction is going to specifically be between products of the gene. So a gene is going to be turned on or go through transcription and translation, and the end product is going to be some sort of protein that was coded for by the gene. And that protein is going to be the subject that is going to interact with another protein at the same allele. So the way that that interaction happens really determines if we get a trait that's dominant or recessive or somewhere in between. All right, so stepping out just a little bit further away from our nice, comfortable relationship of dominance and recessive, we're going to now look at lethal alleles. So, lethal alleles actually cause death at a very early stage of development, usually in gestation. And that usually is going to occur in what we call lethal recessives. So, those genotypes have to be homozygous most of the time to be deadly. A heterozygous genotype or a non lethal homozygous genotype will not be lethal in most cases. So, with these lethal dominant alleles, where we see like a big A and a big A, or a big A and a little a, um, those are gonna usually end up causing death with a very, very late onset. So, we see some of those type of dominant alleles. Um, actually not taking effect until after puberty in some species. All right, so let's look at an example of a lethal allele. This is out of your book, and this is in mice. So, in our parent generation, if we cross two heterozygotes, the mice that are a phenotype of yellow and their genotype is big Y, little y, then they actually have the opportunity to pass on a big Y or a little y at a rate of 50% per parent. So after fertilization has taken place in the F1 generation, we're going to see an obscure ratio happen because a quarter of the offspring actually ended up inheriting the dominant big Y genotype, and they're going to be dead. So we see an alternate pattern of inheritance here in the F1 generation. So we'll end up with 2/3 of the progeny that are heterozygous in yellow, and then a third of the progeny that are going to be homozygous, little y, and non yellow. So if you ever see different kind of odd types of ratios like that, that usually means that there's going to be a lethal recessive somewhere. We can also see lethal recessives happen in some of our plant species. So, snapdragons are a great example of this. So, snapdragons can come in many different colors, but if we end up with a crossing a two parents that are both green, then all of the progeny is going to end up being green. However, if we cross one parent that is yellow with another parent that is green, we end up with a 1-to-1 ratio in the F1 generation from that cross. And all of the yellows will be heterozygotes. So, what ends up happening in this situation is that a big Y is going to be the dominant allele for color, so it's going to provide that yellow color, but it is also a recessive lethal, so that if it shows up as a homozygous dominant form, it's going to be deadly, and those flowers actually won't end up living much past fertilization. So keep in mind that the big Y in snapdragons is going to be dominant for color, but it's also going to be a recessive lethal, meaning that if it shows up in a recessive homozygous form, those individuals are going to most likely die shortly after fertilization. All right, so switching gears a little bit from lethal alleles to multiple alleles, this is where we actually start to see many different types of alleles being expressed. And this is really what creates so much genetic variation in all of our life forms. So, one of the easiest examples for you guys to grasp this concept with is duck feathering patterns. So there's three major feathering patterns that we can see in duck species. So the first one is just a big M with no superscript. That's going to be a wild type, and that's going to be our mallard pattern like we see here. The second allele is going to be an MR, and this is a restricted pattern. So these are what a lot of our female hens actually look like. And then the third allele is going to be a little MD, and this signifies a dusky pattern. And these guys tend to have darker points and a little more definement than the restricted individuals. All right, so if we look at inheritance patterns in our duck species, we're starting with the parent generation first. These are the different types of genotypes that can occur if we were to cross a restricted individual with a mallard. So in our restricted genotypes, we've got three different genotypes that can actually result in a restricted feathering pattern. And there's only two genotypes that can result in a mallard genotype here. So if we were to cross this MRMD restricted hen with an MMD mallard, then they would pass on both of those alleles, and in the F1 generation, we would actually get a 1-to-1 to 1-to-1 ratio from a genotypic standpoint. So we've got a quarter of them that are going to be MRM and a quarter of them that are going to be MRMD. And so, but phenotypically, we end up with a 2-to-1-to-1 ratio. So where we've got 50% of the individuals are actually going to have a phenotype of restricted, 25% are going to have a mallard phenotype, and 25% are going to have a dusky phenotype. So just take some time and work through this with the Punnett square if you need to, but just make sure you have a good understanding of what the restricted, mallard, and dusky phenotypes are. All right, so another example where we see multiple alleles really benefiting the organism is in blood typing. So we have three major types of blood, so A, B, and O. And so keep in mind that some loci or some genes on chromosomes actually have more than two alleles associated with that phenotype. So these are what we call allelic series. And so even though there's more than two alleles that are possible, each individual is only going to express two alleles. So one from mom, one from dad. Okay, so these types of multiple alleles really allow for greater variety when it comes to genotypes out in a greater population. So from those three major blood types, we get different genotypes. So you can be AA, AB, BB, which is B, BO, AO, or double negative. All right, so this chart in your book is actually a really good one. I highly suggest you guys studying it a little bit. So the phenotype of the blood type is shown in this first column, and the genotype that's associated with that phenotype is shown in the second column. And then just to better understand the antibodies that are associated with red blood cells, we've got the antigen type that is carried or coded for by the phenotype or the genotype. So for blood type A, those red blood cells are going to have an A antigen on them. Okay, so they are going to make an antibody for the B type. Okay, so what ends up happening is that if we have a donor that is an A type, then we're not going to get any reaction. If we were to donate blood from an individual with type A, blood into an individual that is also type A. Where we get into major issues is if we were to donate blood and you are a type B and that blood went into an A individual. There would be major reactions because the B blood type actually has A antibodies. So it's going to react very, very quickly. And so we can see those reactions outlined here by the orange. And so that is going to be a major reaction that where blood types are not compatible with one another. So this becomes really important anytime you do blood transfusions and in the hospital. So the way that this plays out is that we end up with the blood type AB, and because both alleles are actually expressed here, no antibodies are going to be present. So these individuals, these AB recipients, can actually accept blood from any donor. It doesn't matter if they're A, B, or O. So because their blood, red blood cells are not going to react with any of the blood types. Where we get into major issues is where we start crossing our blood typing boundaries here that are only expressing one allele. Therefore, they automatically have antibodies to that second allele. So likewise, our O phenotypes, these guys are going to be our universal donors. So these individuals are really important because they can actually donate blood and it can go into any individual that is AB, or O. So type O donors are going to be universal donors, and type AB recipients are going to be universal recipients. All right, so our last slide in this segment here is looking at how to figure out how many phenotypes we can actually get, how many genotypes we can actually get if we're dealing with multiple alleles. So this equation is pretty straightforward. If N is going to represent the number of different alleles on the right hand side of this equation, and N on the left hand side is really determining the number of genotypes with the multiple alleles. So, for example, if we wanted to determine how many different combinations or different genotypes there are in duck feathering patterns, we would know that there are three major phenotypes associated with it, three major alleles. So n would equal 3 here. So we would take 3 plus 1 and then multiply that times 3 again and then divide it by 2 to get 6 different genotypes for duck feathering patterns. So just have a good handle on this equation and how it works and what it will tell you. All right, so we will proceed with section 5.2 in the next segment.